Good evening. I once again welcome all the participants to the last session of the day. It gives me immense pleasure to introduce you to our next speaker, Dr. Samir Srivastav, who would talk and illustrate on the topic interpretation and analysis of biological data. Dr. Samir Srivastav is a senior scientist in Division of Animal Biotechnology (IVRI) in Zatnagar, with over 15 years of research and teaching experience in the area of animal biotechnology. His expertise lies in designing synthesis and characterization of synthesis peptides. He has, to his credit, 2 granted patents on synthetic peptide-based diagnostic. His group has also identified novel AMPs from insect and frog secretion and developed synthetic AMP hydrogel for management of bovine mastitis. I'm pretty sure, sir, your knowledge will greatly benefit all the participants. Now I request Professor S.K. Singh sir to kindly say a few words to Dr. Samir.
Good evening, sir. Can you hear me? Good evening, sir.
Yeah, namaskar.
Namaskar. Sir, it is our pleasure to have you here in this short-term course, and I'm thankful to you for accepting our invitation and giving us time for our short-term course. So I hope your talk will really be beneficial to our participants. So thank you, sir. I think now you can start.
Thank you. So, is my slide visible?
Yes, sir. Visible.
Yeah. So, thank you very much, sir, for this brief introduction and giving me an opportunity to be part of this training program, which I am sure is being attended by several students and faculty members, as I am being told, with a good gathering of audience here. So I'll be presenting my talk on biological data and its collection and analysis. See, we are a veterinary science institute, and I am posted here in the division of veterinary biotechnology. We are doing a lot of work on different aspects of biotechnology and also veterinary science in particular, and in general this institute is a premier veterinary health institute, wherein we are having around 20 different divisions, and the institute is one of the premier institutes in the Asian region. So our main focus is for the health management and production of animals. So we do a lot of research for uplifting the health and production of the animals. And this is how with this brief introduction, I will go straight away to the topic of today's presentation, that is biological data, its collection and analysis. So before I just tell you about the data, I would like to highlight this particular slide which depicts the central dogma of the molecular biology, or the central dogma of the life. So in case of the biological sciences, we all know that there is a flow of information from parents to offspring, from offsprings to their offsprings, and so on. And the carrier of genetic information is the DNA. So
the slides are not. Sir, full slide कर लीजिए sir, full presentation mode में.
So I have done the full screen mode here. So for me it is visible actually.
So visible, sir. Uploaded. Visible, but you have to go to website. ठीक सर, अब ठीक है.
Is it okay now?
Yes, sir. Yes, sir.
Okay. So this DNA is the primary genetic material present in the living organisms, and from DNA ultimately we get proteins through a different process called transcription, translation, and then post-translational modifications. So the transfer of information from DNA to RNA is called transcription, and from RNA we get different proteins through a process called translation. So this is called the forward central dogma, and this is the reverse one, which is also called the reverse transcription. Now we know that there are certain viruses which have the RNA as their genetic material, and still they are able to replicate just like your SARS coronavirus. So it can also go in a reverse transcription mode. There are certain viruses which can make DNA from RNA and so on. So this is the basic principle of biological sciences, where the central dogma of life is the actual flow of information, and all our research in biological sciences mostly focuses on this central dogma. There are groups who are working on the DNA, there are groups who exclusively work on different forms of the RNA, then there is a group who works on the proteins, and of course we are in between of this, the translation and protein part. We are working on amino acids, peptides, and then proteins and so on. So I will briefly touch upon the concept of data, data sets and databases. I'm sure you are already well aware of the data, data sets and databases. I will try to highlight some specific points related to the biological databases and data sets. So why we need databases in the present time? You know, there is if we talk about the 1950s or so, there was very less advancement in the sequencing technologies and high throughput technologies. So the data which was coming out during Watson and Crick's time, when the DNA was discovered, it was very meager, very small amount of the data was available. But with the advancement of so many techniques in biological sciences, it has become possible to generate huge amount of data, and the data comes from different say sequencing technologies, and we have high throughput sequencing technologies in genomics and proteomics. Nowadays another concept is coming up which is looking into metabolomics. Metabolomics is actually the profile of the metabolism of any organism that we are going to see. So in general, we get a lot of data in terms of genomics, proteomics, metabolomics data, then peptide data, then we have certain protein data and so on. So why? Because of generation of such huge amount of the data, it has now become necessary to have some sort of library where we can store this type of the data. So there comes the role of the databases. So there are several recognized databases where the scientists and the researchers get an opportunity to store their data, and some of these databases are freely available. The purpose of the data generation is to make them available to the public and other researchers, so that the duplication of work is avoided. Secondly, the other researchers can get benefited by the research or the findings of the previous workers. So that is why databases are important these days, and we have several of them. I will touch upon few of them in the next slides. So the data which is stored in these databases is useful to the scientists, useful to the computer programmers, useful to the persons who are working in the form of for developing certain softwares. So there are different users for this data. Of course, the major players are the scientists and the students, the computer programmers, the computer scientists, and so on. So likewise, the biological data is maybe of some importance to the industry also, where they are interested in designing some novel probes, novel therapeutics, novel vaccines, and so on. So in general, we have biological databases like sequence databases and structured databases. So
sir, now you can take control. Actually, slides are not changing. At the top, you can click on request or take control.
Just a minute. Yeah. So is it visible now? Is it okay?
Yes, sir. Okay.
Okay. Can you see this cursor? No. Can you see the pointer on the slide? No.
Sir, slide biological database slide is there. We can see that.
You can see the slide. Huh?
Yeah.
Pointer is not visible.
Pointer is not visible. Okay, no problem. So these biological databases, we have the sequence databases where we store the sequences. The sequences may be in the form of the nucleotide sequence or protein sequence, amino acid sequence. And nowadays there is a lot of technology coming up where we are even sequencing the RNA. You might have heard that now the government has taken an initiative to sequence few of the samples which are coming from the COVID testing, just to know what type of mutation it is. So that's what is actually sequencing technology, where we sequence the genome of the virus or bacteria or so on to find out what are the changes in the nucleotides. Now, because this is the RNA virus, it changes very frequently. So that's how the mutations are coming up. You might have heard the gamma, delta and delta plus variants in case of the SARS coronaviruses. So the sequences of different viruses, bacteria and other organisms are submitted in the sequence databases. Now sequence databases can be either the nucleotide sequence comprising the DNA and RNA sequences, or the protein sequences where the amino acid sequences of different proteins are deposited. Similarly, there can be structure databases where the structural forms of the proteins are deposited, which are determined by the biological experiments like X-ray crystallography and so on. So if you see the nucleotide sequence databases, there are primarily the GenBank, then the DNA data bank of Japan and the ENA. Likewise, these are the recognized databases which are publicly available and funded by the European Union or the Japan government or the US government. These are used databases which are getting updated on a daily basis, and here the scientists are free to deposit their the sequencing products, and you will get an accession number from these libraries or the sequence libraries or the sequence databases, and this becomes a public property when you submit the database the data to these databases. Similarly, there is the reference sequence database, and the SNP is also one. The single nucleotide polymorphism database is there. Now this SNP database is, there are certain researchers who are interested in knowing what single nucleotide change makes a difference in the organism. So that is called as the single nucleotide polymorphism. That means at particular site, a single nucleotide may also change, which may result into conferring a particular animal a disease resistant status or making it more vulnerable to the disease likewise. So this is again the description of the nucleotide sequence databases, and these particular databases, the beauty of these databases is that they are updated on a regular basis, on daily basis they are updated, and we have the provision to submit our data either in the form of protein data or the nucleotide data to these databases, and we get accession number which we keep on citing somewhere. Likewise, these are again the retrieval systems, the nucleotide sequence databases, from where you can put a query and search for the different types of the desired sequences. So these are interactive type of the databases where you can search for the nucleotide, or you can search for a particular organism or for a particular protein of any organism. So likewise, these are also retrieval type of the databases. Similarly, in case of the protein databases, there are again primary protein databases where we submit our data in the form of amino acid sequences, and also there are secondary structure protein databases where we submit the protein structures to these particular databases like Swissprot or ProSites. Like in UniProt, we can submit our sequences, and also we can submit the structures. And similarly for the protein information resource, the PIR database, where we can submit our structures and sequences of the proteins. Now here is one example of the protein sequence database where you can retrieve the sequences of different proteins based on the query. Similarly, there are again the tertiary structure databases where the models are submitted. The proteins, if there are certain students who are from the biological background, they might be knowing that the proteins attain different structures, that is the primary structure, secondary, tertiary and quaternary structure, depending upon their functions. The primary structure is just the sequence length of the amino acids. Then when it gets folded, it attains different structures like secondary, tertiary and quaternary structures. So we can submit the different quaternary structures, the tertiary structures of different proteins to these structure databases, wherein these type of structure databases are important to know the interaction between different molecules. Now the most popularity which has gained these days is for the SARS-CoV virus. Many of us might be knowing that there is a protein called spike protein in the SARS coronavirus 2, and it interacts with the ACE2 receptors present in the different say hosts. So how people have understood that there is interaction, because the ACE2 receptor sequence is already available, the secondary structures are available in the database, and the sequence of the spike protein was submitted, the secondary structure was generated, and based on the interaction, the 3D interaction was studied in silico, and it was found that the ACE2 receptor binds very strongly with the spike protein of the coronaviruses. Now based on these findings, the research has further aggravated, and now I was just seeing a paper. People have found out that in case of the bats which harbor the SARS coronavirus 2, these bats actually have a spike protein, have a ACE2 receptor which binds slightly loosely to the spike protein of the coronavirus. That may be one of the reasons that the bats doesn't get affected with the coronavirus, although they harbor the virus very efficiently and they become the transmitters of the virus. So based on these studies available with the bioinformatics tools, the researchers are greatly benefited. This is one example of the secondary structure and the tertiary structure databases. So towards the left side, you can see a plot which is called the Ramchandran plot, based on G.N. Ramchandran of our country, who has given who has done a lot of research on proteins and proteomics, and they have found out that there is a allowed and disallowed conformation based on the structures of the protein, the secondary structures of the protein. So you can generate a Ramchandran plot from these type of the databases, where you can submit your protein sequence and you will find out a Ramchandran plot. Similarly, toward the right side, you can see the tertiary structure of the protein, where you can see different helices and beta sheets and turns. So this is how the protein is represented in case of the structure, the 3D structure in silico. Coming on to some information about the work that we are doing. We have a specialization of making the peptides in our laboratory. Now peptides, you can say that the peptides are nothing but they are the chains of amino acids. There are 20 different standard amino acids in the biological system. Of course, there are other known standard amino acids also present in the plants, fungi, bacteria and viruses. These 20 amino acids are say combined together in different patterns to give different proteins. So when the two amino acids join together, there is a removal of one molecule of water and formation of the peptide bond. So the two amino acids can combine together by removal of one molecule of water and formation of a peptide bond. Thus we get a dipeptide. Similarly, the tripeptide, tetrapeptide, pentapeptide and so on. So as and when the amino acids join together, you get the peptide chain, which may be from two amino acids to several amino acids, and this is how we generate the peptides. In case of the biological system, the peptide synthesis or the protein synthesis is a process wherein there is involvement of lot of infrastructure and machinery of the host or the body. Different enzymes are involved, and thus we generate different proteins. But in our laboratory, we are doing it through a process of chemistry. We have 20 different boxes containing different amino acid derivatives, and we have a very well defined chemistry which is called the solid phase peptide synthesis chemistry, which was developed by Bruce Merrifield in 1963. And this particular scientist got a Nobel Prize for this particular discovery of making of peptides using two amino acids in 1984. So This particular technique is now very well developed, and a lot of industries are running on peptide synthesis. We have a specialization of making different types of the peptides in our laboratory, and we do a lot of data generation based on peptide synthesis and the application of these peptides for different biological applications. So with this small background on the peptides and proteins, I will just take a small tour of yours to how we have joined hands with IIT BHU and how we have come across some work wherein we have used the biological data available in the databases and developed certain softwares or certain algorithms using artificial intelligence, the group led by Professor S.K. Singh Saab. So this is the peptide bond I already told. Then the two peptide bonds can be joined together using certain intrapeptide bonds or interpeptide bonds. That's how the proteins get folded in the solution. Now, coming on to the different say aspects of the peptide, different researches what we are doing in the peptide area. As I already told you that there are 20 different amino acids. So in terms of mathematics, if you can see, we can generate the diversity of 20 to the power 20 if we replace different amino acids. So that is the diversity which is involved in synthesis of the peptides. Suppose if you are making a 10 amino acid length peptide, we can generate a diversity. We can change each of the 10 amino acids with 20 different amino acids at each 10 positions, and you can imagine what type of the diversity or the data that we will generate when we work in this type of the libraries. So there are different say areas where we are working, where we are touching upon. There is an area of receptor ligand interactions, as what I have given an example of spike protein and the ACE2 receptor. So basically your ACE2 receptor is a protein and the spike protein is also a proteinaceous molecule. So this is a protein-protein interaction. Similarly, there is a concept of enzyme and substrate. So there are enzymes which acts on the substrate and convert it into products, and that's how you get different say biological reactions taking place in your body. There are certain hormones which are peptide hormones, and they play a very important role, like your the vasopressin or the insulin. You might have heard the insulin. This is a peptide hormone which controls the blood sugar in the body. Then there are certain venoms in the molecule, in the organisms. The easiest one I will tell you is the snake venom, and the venom of the snake contains a lot of peptides which are toxic in nature, and there are certain peptides which block the receptors. That's why when there is a snake bite, you will the patient will feel paralysis or will have symptoms of inflammation and so on. Then there are toxins. Those of you who might have experienced the bite of the wasp might very well understand this. The sting of the wasp contains certain toxins which are peptide toxins, and once these wasp stings on the body, these toxins are injected into the body and they actually block the adrenergic receptors and neuronal receptors, and that's why you see a lot of allergic reactions taking place. Then there are certain anti-inflammatory molecules which are peptides which prevents the inflammation. The body has a mechanism of defending itself, so it will always try to prevent inflammation caused due to the invading agents. There are certain defense molecules that I will deal in more detail because we are working on the host defense molecules. There are certain antimicrobial peptides which are secreted by the hosts or the organisms, and these particular defense molecules actually play a role in protecting ourselves from the attack of different bacteria or viruses or fungal or so on. Then there are regulators of body functions. I have already told that there are hormones, there are certain cytokines which are playing a role in regulating the body functions. Now in our laboratory we are working on these areas which are given towards the right, like antimicrobial peptides, the cell penetrating peptides, the cell adhesion peptides, which are actually used for developing the biomaterials, where we plan to develop certain materials which can heal the wound quickly. Then we are also working on tumor targeting peptides, so we can tag the anti-tumor drugs to these peptides and we can carry these drugs to the the tumor cells. Then we are also working on plant defensins, wound healing peptides, and certain immunogenic peptides. So these are different areas which our students and our group is working in this area of synthetic peptide biology. Now here I would like to thank the IIT BHU group, specifically the computer science engineering group of Professor S.K. Singh Sahab, where we have joined hands with this group for last 3 years or so, and we are hand holding the expertise of this group in doing certain computer based studies, and we are taking up the work on the lab scale. So our combined efforts with a wet lab and dry lab experimentation has given us dividends, which is quite visible in our publications and also projects that we are handling and also in pipeline. So basically we need computer scientists or computer engineers who can handle the data, who can develop the models, who can change the algorithm based on our requirement to find out novel molecules. And we as a wet lab researchers, what we can do is we can make the molecules in the laboratory, we can synthesize the molecule, we can do recombinant DNA work, produce a lot of recombinant proteins in the laboratory to develop certain molecules which can be tested in the lab and develop certain drugs or develop certain say vaccines or so on. So it is basically a handshake between the two groups to develop some biological molecules, and mind it that all these processes generates data and also handles data. So that is what is the relevance of today's talk, that these particular researches are possible only when we are able to handle the data using the supercomputers available at your IIT BHU group, or the servers and the online servers. There are certain programs which are available online. So these particular servers and the computer programs are used to handle the data and to generate new data. And such molecules which are identified by these groups, when they are given to the wet lab researchers like us, we have the capability of testing these particular molecules in the laboratory and coming out with some novel compounds like drugs or the antimicrobial molecules and so on. I will touch upon two different aspects which we have recently covered and published papers in this particular area, where we have handled a lot of data and generated the algorithms and servers to handle the data in a different way. So this is the first part, which is called the ABPpred. I will just we have done two work, so I will show you one work first. So herein we are working on antimicrobial peptides. So this particular work is for generating new antimicrobial molecules. Now to introduce you about the antimicrobial peptides, I will just tell you a brief introduction which will make you understand how and what these antimicrobial peptides are actually. So if you see, the discovery of the antimicrobial peptides is around 20-25 years back, just with a basic observation that the frogs, you might have seen the frog lives in different dirty environments, in different say mud or in all sorts of environment which is full of the bacteria. So there was a scientist called Lagerhoff, and he found that their group found that these particular frogs do not suffer from bacterial infections as we suffer, the humans or the higher organisms suffer from more bacterial infections. They found that these particular, the Jasloff and Lagerlof, these two scientists, they found that these particular frogs, even though they are living in very dirty environments, still they are resistant to a certain extent to the bacterial attack. So they were say fascinated by this particular observation, and to find out what could be the reason, what they have done is they have taken the frog, they have cleaned their surface from outside thoroughly, and then they have put these frogs in different solutions containing pathogenic bacteria, very highly pathogenic bacteria which causes disease. So in those bacterial solutions they have put this frog, and then they have found that whether the bacteria suffers from disease or the frog suffers from some disease or not. So to their surprise, they were amazed to see that the bacteria doesn't infect the frogs. So they were thinking that the intact skin of the frog might be the reason, that because of the intact skin the bacteria is not entering. So what they have done in the second experiment, they have put the cuts on the skin, they have created the bruises on the skin, and then again they have put these frogs in the bacterial suspensions. Then again they found that there was almost no bacterial infection in the frog. So this gave an insight to think that there must be something in these particular frogs which they might be secreting, or there must be a mechanism by which these frogs are preventing the bacterial attack. So from there they could discover the antimicrobial peptides by a tedious process. What they have done is they have removed the skin of the frog, and then they have triturated the skin. They have done a lot of biochemical analysis and isolated certain compounds from the skin secretions, and which they named them as antimicrobial peptides. And they found that these were small molecules of maybe 20, 25, 30 amino acids long, which are secreted in response to the bacterial attack. And because of these antimicrobial molecules, the frogs are ultimately protected from the attack of the bacteria. So this was the basic information which people generated about 30 years back. Until the AI came into place, till the computer algorithms were developed, what people were doing is to identify the new antimicrobial peptides. They were just catching hold of these frogs, giving them a DC shock. They were giving a DC current shock to these frogs, then collecting their secretion. Because of this DC shock, the pores of the skin of the frog opens and releases the secretion outside. So the scientists used to collect these secretions and do lot of analysis in the laboratory to find out what new molecule is secreted by the frog. And that's how the discovery of the antimicrobial peptides went on. And these particular sequences of these antimicrobial peptides people used to deposit in different databases, which are called AMP database. Nowadays there are more than 20, say 15-20 databases available, which are updated on a daily basis when new sequences are submitted to these databases. So now we can say that there are around 4,500 sequences of AMPs available in these databases, which people have identified from frogs or from insect or from the spiders or from scorpion or from other different organisms, even plants. So this is the basis of our work. Now what we did is, in our previous work, when we have not come across with the IIT BSU people, we were also doing the same thing. We had a tie-up with people the scientists in Kerala, and they were catching hold of the frog in the forest and collecting the secretions, doing a lot of biochemical analysis and sending us the sequence. So in our lab we used to synthesize these peptides based on the amino acid sequence, and based on this peptide that we synthesized, we were actually doing a lot of biological experiments here. And you can imagine how tedious it was, how tedious work it was to catch hold of the frog in the environment, in forest, and then collect the secretions, do all biochemical tests and so on. So with this particular group, what we did is we applied artificial intelligence here, and we developed a few models by training our models using the data available in the dataset, in the databases. We created two different datasets, which is called as the AMP dataset and the non-AMP dataset. So practically we took the advantage of the data which is available in the databases. Yeah, so the data which is available in the databases, we made a thorough search of different AMPs which are present in the databases, and we created the different data sets of AMPs and non-AMPs, and then we developed the AI machine learning and deep learning based models, trained our models using the students and the researchers of computer science engineering group at IIT BHU, and then we developed this robust model for predicting the novel antimicrobial peptides. So now through these models, what we can do is we just have to put the protein sequence into these servers, and we can get the probable sequence of the new antimicrobial peptides based on the learning of the machine learning and deep learning methods which the computer has learned based on the data sets already available. So this particular work we have published in different journals. So this is actually a brief summary of the work that we did. We compared our model with the other say existing models which are available in the public domain, and we could find that there was a good sensitivity and specificity of our model and different diagnostic sensitivity parameters we have seen. Then we have used a set of data for independent evaluation of our model with the existing model. So I already told you that there are say 7 or 8 different models already available in the public domain which do the same job. Of course, there are certain strengths and weaknesses of each model. So we tried to overcome the weaknesses of the previous model and develop a robust model which performed better than the previous models. Then we have compared our results with the different data sets using our validation data set and also the test data sets, and we compared the performance using the ROC curves. And then toward the end of this particular work, what we did is we identified new antimicrobial peptides in the proteins of bacteriophages. So if you can see this figure in this particular slide, this is actually a virus which infects the bacteria. That's why it is called a bacteriophage. That means a virus which kills the bacteria. So we thought that because these particular viruses are killing the bacteria and the mechanism is almost similar to the mechanism of antimicrobial peptides, so we thought that these particular bacteriophages could be a source of novel antimicrobial peptides. That's why we explored certain proteins of these bacteriophages, and you can see in these particular slides we have selected one particular type of the bacteriophage which infects the bacteria called Streptococcus, and we have generated different say around 10 million different peptides, and based on these particular models we have identified few of them, and out of this 10 we have tested on the bacteria and found very good results that these particular antimicrobial peptides which were identified using the artificial intelligence tools were actually able to inhibit the growth of bacteria in the laboratory. So practically this particular work is a complete work where you can say that it is from from dry lab identification of the molecules to wet lab synthesis of the molecules and then testing of the molecules in the laboratory, and now we are trying to take few of these molecules to the field conditions where we can use these particular antimicrobial peptides for treating conditions like burn wounds or open wounds. You can say that there are several antibiotic creams are available which are used for treatment of the wounds so that bacterial infection doesn't occur. So we would like to use these antimicrobial peptides for treating these conditions. And this is the web server which is deployed from this particular work, wherein the users can submit their the protein sequence and get the AMPs as output. So this work was published in Briefings in Bioinformatics in March this year. Similarly, the second work also is similar to this, wherein we were interested in locating or identifying the host defense proteins. Now here again, briefly I will give the background in 1 or 2 minutes. The host defense proteins are the proteins which protects the organism from invading microbes, just like the antimicrobial peptides. In case of the smaller organisms or lower organisms like insects or frogs or lower vertebrates or fishes, they are the antimicrobial peptides. And in case of the higher organisms like us or the other mammals, they are called the host defense proteins. And primarily they are, although they are also called antimicrobial proteins, but they are in general called as the host defense proteins. And there are different categories like defensins, like cathelicidins, like mesins. Likewise, there are different names by which these host defense proteins are designated. But you can say one example I will tell you. Just see the secretions from the eye or the tears which are coming in case if some dirt goes into your eyes, you will find that there is a secretion of the tears. Now these tiers also contain certain antimicrobial peptides or antimicrobial proteins. So the purpose is that because your eye is in direct contact with the environment, likewise your nose or the mouth saliva or ear, which are directly in contact with the environment and constantly come across the bacterial infection. So there are certain proteins at these particular sites which actually protects your body from invading bacteria by killing the bacteria non-specifically. So these are called the host defense proteins, and we aimed to identify novel host defense protein. Although a lot of host defense proteins are already available in the databases, so we wanted to identify the novel or the new host defense proteins in the genomes of different organisms. That's why we took up this study with this IT group. So initially what we did is we created our own data set, and for creation of our data set what we have done is we have taken into consideration all the data available in the public domain. We classified the data into different categories. We have taken into category all the available phylums of the animal kingdom. So this is for the animal kingdom. So you can say that the animal kingdom has different phylums. So there is and then they are divided into different classes like insects or fishes or mammals or spiders or snake likewise. So we first identified how many sequences are available for each particular class, and then we created our data set. Then we developed models for say training, testing and validation of our data, and then after a robust model was generated, we compared our performance with the existing few of them which are existing in the public domain, and to find out the diagnostic effectiveness parameters. We evaluated our data using the validation and the test data sets, and also the performance was validated using this external or the independent data set, and also the performance was say depicted in the form of the ROC curves. We could see that our model, which was developed and trained using all the different available sequences, showed good performance. So here you can very well appreciate a comparison of different 5 or maybe 7 different models, 7 or 8 different models which are available. So in the lower say middle, the lower row in the middle portion, you can see this is our model ABPpred. So this actually density plot gives an idea that if we see the AMPs and non-AMPs, how correctly your model predicts the non-AMP as non-AMPs and AMPs as AMPs. So if there is very accurate prediction, you will get 2 different peaks at different points, one for the AMP and one for the non-AMP. And if there is some lacuna in prediction, or if it is predicting with lower accuracy, you can see the merge curves, as is very well evident in other models. So you can say that there is overlap of the AMPs and non-AMP data. So some of the AMPs are designated as non-AMP. Similarly, certain non-AMPs are designated as AMPs. So we can find that our model was doing pretty well in this particular thing. So having done this, we were interested in predicting the novel antibacterial molecules or the novel antibacterial proteins in the genome of different animals. So we had taken one representative example, which is a annelid, a small molecule or a small say organism, which is a leech. It belongs to the class of leeches, and the genome of this leech was available in the public domain, but most of the proteins were not assigned any function in this case. So we have selected this particular genome, and using our model we could find that we could identify 500 or more different say antimicrobial or the probable antimicrobial proteins. And when we have seen the similarity of these predicted antimicrobial proteins, we could find that they were actually matching with the antimicrobial proteins of different species. So that gives a confidence in us that our model is working fine. So whatever the molecules or the proteins that we identified using this particular say algorithm were actually matching with the different antimicrobial proteins or host different proteins of different species. So this we did for different proteins, 3-4 proteins. And we developed a web server for this particular work also, which is freely available on the public domain. And you can just put your protein sequences, and it will give you a probability score of the protein being antimicrobial protein or not. So this work was also published in Briefings in Bioinformatics maybe last week. So a quick look on another 10 minutes I'll take what other type of the data can be generated with the help of the computer science. There is a new concept coming up, AI or Internet of Things in animal agriculture, both in agriculture and in animal science. So this is how the people are looking towards future. We would like to integrate the computer science and Internet of Things for tracking the disease spread, for early diagnosis of diseases, for prediction of the onset of diseases. You can say that at particular seasons there is an onset of certain diseases. Like if you see Japanese encephalitis, there is a seasonal occurrence of Japanese encephalitis in particular areas. So if we can use the artificial intelligence machine learning tools, we can predict the onset of diseases. So that is the concept of Internet of Things. Another very important concept is sharing of the research data. So we know that there are different groups working on different problems, and using this Internet of Things we can very well share the research data by putting it in cloud or in a particular database or servers, and likewise we can do disease monitoring, and then very importantly we can monitor the response of patients to particular drug or therapy or cancer treatment, likewise. And another very important point is networking of the specialists. Now this is very important. If you see the Medanta group of hospitals are there now, there are several Medanta hospitals throughout the country, or Apollo chain of hospitals, or likewise. So if you are live posted, or if you are admitted in Delhi and the expert of the neurology is available in Gurgaon, so if there is Internet of Things type of the connection network, you can very well link up the neurosurgeon from the Medanta group in Gurgaon, and he can give advice to the patient admitted in Delhi or remote place like Jharkhand or anywhere else. So this is like networking of the specialists. This is one very important thing which has happened by say networking of the specialists. Likewise, the similar thing we would like to take up in the animal sector also. During this COVID outbreak, this particular Internet of Things has given us a bright example where the specialists and the researchers throughout the world are now connected, and the WHO and the OIE are collecting the data on daily basis for the emergence of new viruses, the spread of the new viruses, and so on. So this is the beauty of the data management integrated with the internet and the computer scientists. So we can have a controlling of the diseases and disease prevention. We can hook up the diagnostic services. We can have a smartphone connectivity wherein the farmers can just take a snapshot of the disease condition of the animal, put it in a particular server, and it can go to the expert, and the expert can just see the symptoms or just see the video and give a tentative diagnosis that these particular symptoms can be of this particular disease. So you take these measures, so and inform the disease specialist accordingly. So we can save the animal, or we can save the spread of especially the contagious diseases like COVID or the other viral diseases. So the another very important thing is the automation of the farms and the livestock health management. So this has been done in a big way in countries like China or Europe or many other countries where the dairy farms are now almost fully automated using the sensor network and using the Internet of Things. The movement of the animal is monitored, the health of the animal is monitored, the different activities at the farm is monitored through a network of different say computer networks. And this is the concept of the smart farms, which is very well coming up in different countries. We also wish to integrate the computer science people and the sensor networks in our livestock farm in our institute. So we have already submitted one project. If it goes through, we will definitely have some fruitful outcome in managing the farm in a paperless way, the livestock farms. So you can also monitor the different parameters like the body temperature, the secretions coming from the nose or the eyes or so on using the sensor networks. This is the automated milking shed you can plan, wherein you can record the milk yield. You can have the milk quality monitoring. You can also see if the milk yield is appropriate. You can also hook up with certain automated machines where you can find out if there is any residue of the antibiotic or the pesticides and so on. Likewise, in case of the pet animals also, there are pet collars or wearable sensors coming up where you can put certain collars to the pet animals, which prevents the insects away from these animals, which give you an idea about the different type of the physiological conditions of the pets, and also track the movement. In fact, these particular wearable sensors have found a lot of applications in wildlife and zoo animals. In case of the wildlife, you might have heard about the tracer techniques where the lion or the tigers are just put with the radio tracers so that we can track the movement of the tigers or lion or the wildlife populations. Similarly, for the migratory birds, they have put the GPS tags so that you can monitor the real-time monitoring of the movement of the birds can be done. So basically, all these things which I have told you till date or till now, these all generates a lot of data either in the form of images or in the form of the nucleotide sequence or the structures, or in the form of the peptide data or the protein data, nucleotide data, or the data of the health specialist and so on. So these particular data found their place in libraries like databases which are publicly available, or like servers which we have developed in our papers and so on. So these are the different applications where you can find the data and you can retrieve the data in an interactive way and also do different applications for your research. So this is our strength. If somebody in the group wants to collaborate with us or with join hands with our group with IIT people, this is our strength. I will take only 1 minute here. We have, as a pioneer institute in veterinary science, we are having lot of clinical samples available in the polyclinics and the hospitals. We have the disease diagnosis data available with us. We do a lot of nucleotide sequences, protein sequencing, lot of peptide work. We have a lot of histopathological images. We are working on animal cancers. We do a lot of work on bacteria and viral diseases, fungal diseases. We do a lot of clinical diagnosis using ECGs, ultrasound, X-rays, and MRIs and so on. So we do a lot of work on genomic studies, interaction studies, and certain basic sciences. So the possible areas of collaboration can be developing the smartphone-based applications for monitoring the health of animals, for tracking the animals, for managing the farms. We can work upon using the AI tools for data management and predictions. We can give the livestock farm management solutions using different computer experts. We can also develop certain mechanisms for health monitoring of the animals, and very importantly, we can join hands for better teaching and training of the students and researchers who are aspiring to do research in the area of artificial intelligence, machine learning, or the Internet of Things. So basically, we are doing a lot of interdisciplinary work within institutes, with different institutes, and now we are open for the transdisciplinary research and collaborations with people like IIT or computer science or electronics or sensor groups and so on. So with this, I thank you for your patient hearing. I thank Professor S.K. Singh sir and their group for giving me an opportunity to share our work and also to interact with this vast group of participants in this training program. So if there are quick questions, I will take up. Otherwise, we have our team members, Ritesh and Vishakha and Abhinav, to take up the other questions, please. Thank you very much.
If you have any questions, please raise your hands. Okay. I think, sir, there are no questions.
Okay.
Okay, there is one question. Sir, would you like to take it?
Yeah, sure. Where can I find the question?
Sir, I will help you with it.
Yeah.
Akash, please unmute yourself and ask your question, and please be quick about it.
Hello, am I audible?
Yes, you are
please.
audible.
Yeah. So thank you, sir, for the kind presentation. Actually, in one of the slides, you had mentioned you were using bidirectional LSTM. So I just wanted to understand, like, is it the state-of-the-art model? Because there are plenty of the models in deep learning, right?
Yeah. So I would
Akash.
pass on this question to Ritesh, Vishaka, and Abhinav if they can interact and answer.
Kindly be loud. We are not able to hear. Kindly be loud.
Am I audible?
Akash, actually, BiLSTM is a type of RNN. RNN means recurrent neural networks. It is a type of deep learning algorithm that is used for sequence-based data. Okay, for like tasks like natural language processing and all. So we can use this for classifying peptide sequences as well. Okay. So this is a model used in deep learning.
Okay.
So in
You
addition
can
to
refer
that,
these publications. If you have any questions, you can query us on email. Okay.
Okay.
Ankit? Okay. Thank you, Dr. Srivastava, for such an insightful and intellectual session on the interpretation and analysis of biological data. This lecture witnessed an active participation of more than 500 participants. And personally speaking, this lecture was sort of very helpful in my ongoing research. I'm sure our audience too must have gained a lot of knowledge from this lecture. And we would be glad to have you with us in such future events as well. And to express our gratitude, we would like to present you with a virtual memento. And I would like to invite Dr. Singh again, Professor Singh again, to say a word of thanks to Dr. Srivastava.
Thank you, sir, for giving us such an informative lecture to our participants. And I hope this your lecture must be very motivating for the participants to work in the area of this veterinary science and this antimicrobial peptides and other areas that you have shown. So I hope some of the participants who would be interested in doing the work, they'll send the mail and we'll communicate to you.
Yes, sure, sir. Yes. Thank you very much, sir.
Thank you, sir. Thank you, sir.